Our next speaker is Mo. He's an associate professor at pathology and bioengineering here in Pittsburgh. He performed his postdoc in MIT in synthetic morphogenesis and organoids, and he will be talking about a novel human embryo body that shows that they can get like the first blood transformation. So Mo, please.
Well, thanks so much for the invitation. Thanks all of the organizers and you all for giving me the honor of presenting some of our studies here today in the context of modeling post-implantation human embryogenesis. And I would like to start with this slide that a single cell managed to create one of the most complex living systems that we know of, 37 trillion of cells and more than 200 cell types. And it does it with a high degree of reproducibility and robustness in form of cellular proliferation, cell migration, multilineage communication that emerge into the form of the tissues and organs. And it does it with a high efficiency and reproducibility. But when we start to do that in the lab, when we start to make tissues and organs for transplantation, for example, we start to lose that robustness, and we don't know why. So that is one of the central questions when we think about building tissues and organs for regenerative medicine. At the center of this process of embryogenesis, we have this post-implantation, early post-implantation part of development, that embryo starts to embed itself in uterine wall. And it's kind of hidden from our access for both practical and technical reasons, but many, many important events happen at that stage of life, such as blood formation, access formation, we have development of the different organ buds, and the development of the nervous system or neurulation. So many tissues and organs generated at that first month of development, and then we have more growth and maturation after that. So understanding that stage of life would be quite important, and this is an embryo at that stage of implantation. You can see here how small it is, and it is obvious that would be very hard to study that when it is hidden in the uterine wall. So this is one of the reasons that there are very interesting sentences about this stage of life and the events that happen. For example, it was said that it's not birth or marriage or death, but gastrulation, which is a truly most important stage of your life. And when we look at post-implantation stage and compare it with the other species, we can see a huge difference. So this is also generate additional motivation why we should study human. And also we know that many pregnancies, 60% of pregnancies would fail during this peri-implantation and prior to that implantation phase. So because of all of these, we need models to be able to study post-implantation human embryogenesis and thinking about what could be those type of models. What are the approaches that can provide us with appropriate models to study and capture complexity of human. And model is not exactly the same thing, but it's a close reflection. It can come with a different variety. It can report on the reality in a different version, sometimes augmented, sometimes approximate. In some cases, can capture some of the events much more and very close to reality. And it should be efficient, accurate, tractable, and ethical in this case. And I'm going to give you two different kind of stories of one embryo model that we have recently developed in a lab to study post-implantation. And then also starting to think about, in a second story, I'm going to kind of initiate this thought process of how we can use these embryo models with a variety of the applications, not only focus on reproductive biology. So this is, you have heard about this slide several times today. You can see embryo would start to develop. We have this plus to cis stage that embeds itself in the uterine wall, and we have three different parts here. We have extraembryonic part, that is trophoblast. Here we have another extraembryonic part, which is named primitive endoderm. Hypoblast would give rise to yolk sac eventually. And we have this red cells embryonic part that's going to give us the future embryo. What Josh Hislop, one of the graduate students, approached in the lab, he used induced pluripotent stem cells and focused on these two layers. One layer would be these wild-type cells that are going to generate epiblast, and then another engineered cell population with this transcription factor to be able to mimic the formation of primitive endoderm. I'm not going to go through the details. We mix these two cells through specific conditions, and we look at them, what will happen in cultures. After 5 days, we can get these type of structures, and these structures are similar to the embryo attached to the uterine wall in which the yolk sac is cut open and expand on a dish. We don't have a trophoblast layer, so it actually provides us with ability to eliminate potential of these structures, and we would be able to follow them over the time with the minimal ethical challenges. And we have studied the interaction of these two layers of the hypoblast extraembryonic endoderm and the epiblast layer. You can see every one of these structures, these red cells are red spheroids are this part. From the lateral view, we have a small cavity in them, and then we have this green layer here that expand and confine these structures. And I'm going to show you that this is a yolk sac tissue. So when we start to make this in the lab, it's going to provide us ability to study human events in culture. As we have seen today, being able to observe these events will provide us with a powerful way to see how cells make decisions, how they start to decide their fate, how they self-organize and make structures, and many developmental events actually discovered by observing these kind of behaviors in different model organisms. And here, I'm going to show you some of these events as an example. For example, when we mix these two cell types, they start to sort from each other. They make these embryonic discs. They start to generate this vertical growth. After that, we start to get proliferation of the cells, migration, and here you can see how the cells start to interact with each other. And interestingly, as a result of this interaction and production of the specific molecules, we start to see events that result in a cavity formation. So now we are basically seeing how human cells make decisions to make a cavity. And when we study this cavity and we start to see what are these cavities, we start to see that these are very similar to amnion cavities. We see that the layer of the cells attached to the dish has chosen to become amnion layer, and then we have two layers of the cells on the top that make bilaminar discs. I'm not going to go to the details, but I want to show you an example of how now we can see events that happen in actual embryo. We can capture some of them in culture in a dish that provide us with a human model to study human embryogenesis. Here, formation of the posterior part of the embryo marked with this marker TBXT, and we can see that these embryoids, this cluster of the cells, they are not just homogeneous population of the cells, rather than they make this symmetry breaking. They have access in them, so it's kind of provide us again with a way to study access formation in human. It doesn't happen in all of them, it happens in a portion of them, which is also important because we can start to compare failed versus the successful events and then make conclusion why things can go wrong during the embryogenesis. But I want to focus on another part of these cultures, which I showed you initially, the green layer of the cells that is going to give rise to the yolk sac. And yolk sac is an important entity with multi-organ function during development. One of the important part is development of these blood islands that give rise to blood formation. And also, it doesn't just generate a transient wave of blood, rather than it generate cells that they can migrate to the embryo, and they stay with us for the whole life, such as macrogelia, for example. So you can also imagine early events in life can influence later stages, how macrogelia, for example, behave in a brain. When we follow this culture over the time, we start to see generation of these round cells budding up from the culture. If we stain this culture for other markers, we can see these cells bud up from these endothelial vessels. And when we look at them more closely in 3D, we can see that these cultures have these endothelial tubes, and these yellow cells are actually in these tubes, generating potentially from the endothelial cells, kind of starting to teach us about how blood is formed for the first time in the yolk sac and what type of blood may be formed, and we are starting to further gain understanding of how these neighborhoods are self-organizing yolk sac. For example, we see in a neighborhood in the yolk sac that it is really good in generation of platelets, or a neighborhood that can generate red blood cells or macrophages. So now provide us with a human-based model to study events at earlier stages of post-implantation when it is very hard to access actual embryos. And now I'm going to kind of switch gear and give you an example, just a tip of the iceberg of the potentials of these models. When we start to develop these models, we provide an opportunity to access to the cell type, niches, and events that are quite hidden from our access. And they're obscure part of the development. And one of the key process that we think in the lab is that they are not just important for reproductive biology, but they have far more impact. They can provide us with access to cells that are important for regenerative medicine. And we know that hematopoietic stem cells stand on a very important therapy for a variety of diseases, such as genetic disease, cancer, and also infectious disease. But hematopoietic stem cells are limited. We have a limited access to matched donors. When we harvest them in the lab, when we want to keep them and expand them outside the body, they lose the differentiation. And there's also racial disparity in the hematopoietic stem cell access. For example, in blood banks, we have a different distribution of these cells in the context of what population would have access to matched hematopoietic HS, or HSCs. And here, one of the goals that we pursued in the lab was, can we use this yolk sac tissue as a way for providing a near physiological support for keeping hematopoietic stem cells alive outside our body, making them to stay more in a more stem cell state, and we expand them. And we use cord blood hematopoietic stem cells. This is just a few example slide of what we did. We co-cultured these hematopoietic stem cells here with a niche that is our yolk sac niche. And you can see here, when we add these cells on top of the niche, they start to gradually expand and interact with the niche. Then we harvested these cells and implanted in the mouse, and you can see here, the cells that are in contact with the niche has shown much powerful ability to reconstitute immunodeficient mouse and produce a multilineage human cells in the immunodeficient mouse. And this is the size of the spleen that start to become much larger, showing this reconstitution. And the ability of the niche to produce all of this growth factor actually helped us to develop augmented function, higher quantity of the hematopoietic stem cells ex vivo, ex utero outside our body, and also decreased cost substantially. So this is a very clear example of how these embryo models can help regenerative medicine. We are also actually starting a startup from this part of the studies to translate that into patients. And in summary, first I want to show you how engineering iPS cells and then combining them in a right way start to capture parts of the development that can capitulate the post-implantation stages of life. We could generate these hexembryoids that are very similar to the a flattened yolk sac with a hypoblast-epiblast interface and amniotic cavity. And also the fact that the embryo modeling would help us not only for reproductive biology, for understanding why pregnancies fail, and starting to study underlying genetic diseases of, in a context of human development, but also provide us with this access, very valuable access to cells, niches that help us for our approaches in regenerative medicine. And with this, I would like to thank my lab, collaborators, and thank you for your attention.